This is video 5 of our photosynthesis lecture series, and this one will focus on the dark reactions or the light-independent reactions, also called carbon fixation or Calvin cycle. Alright, so these are the reactions that happen in the stroma of the chloroplast. So this is outside the thylakoids. It uses the energy produced during the light reactions, which we discussed in videos 3 and 4, and using the electron carriers produced during the light reactions, carbon will be fixated or trapped into glucose, and that's basically the point of this. Now, some people call it dark reaction because it doesn't need light, but I also learned to point out that I don't like that name because it makes people think that it only happens in the dark. But this process can actually happen during the day or during the night. Plants are capable of doing it at both times. It just means it doesn't need light. Although that's actually also misleading because you do need light. Because if you don't do the light reactions, you can't do the dark reactions. The only difference is that it can happen during the night using the energy produced during the day through the light reactions. So while plants will do this process during the day, it will continue during the night using all the energy that was produced during the day. So the daytime produces plenty of energy to be used during the day and then some, which throughout the night the plants continue to use to continue photosynthesis. So plants will continue to trap carbon dioxide even into the night using the energy that they have stored from the light reactions during the day. So what actually happens during these light-dependent reactions is that carbon dioxide is trapped from the atmosphere into molecules like glucose. Now, glucose is a higher energy state, so that means this is a reduction reaction where electrons are stored. Of course, because remember, the overall picture is that electrons are going from water to carbon dioxide, so while water is getting oxidized, carbon dioxide is getting reduced. So, and that's what you see here. The electrons of water are replenishing the electrons lost by the photosystems as the photosystems get hit by light to charge the electron carriers, which are then used to power up the trapping of carbon dioxide and to make sugar. So this is the overall picture, and we're going to be discussing exactly how this process takes place. So again, this happens in the stroma, and it's called a Calvin cycle after the scientists that discovered it, but some people also call it carbon fixation because it's trapping carbon from the atmosphere. So this is all made possible by a molecule called Rubisco. Now basically the way Rubisco works is that it has a high affinity for carbon dioxide, and whenever carbon dioxide gets close to Rubisco traps it, attaches itself to it. Now Rubisco is actually a 5-carbon molecule, so when the carbon dioxide attaches to it, it immediately makes a 6-carbon molecule. But this 6-carbon molecule is a little bit unstable and it can't survive very long, so almost immediately it splits up into 2 molecules called 3-phosphoglycerate, also affectionately named or abbreviated for the love of God, because it's very hard. We call it 3PGs, basically. 3PGs, so 3PG, or 3-phosphoglycerate. Now, these 3PG molecules do have a phosphate group in them because Rubisco has a phosphate group in them. So, you notice that. Now, since you had a 6-carbon molecule here, now each one of these molecules will have 3 carbons, and you see that in black in this drawing, that each molecule has 3 carbons. Now, the very next thing that's going to happen with the help of enzymes, ATP produced during the light reaction process through proton motive force in that ATP synthetase turbine is going to be consumed. ATP is going to give up one of its phosphate groups, and that phosphate group is going to be added here to create a molecule that looks alike like 3GP, but it's charged, and this molecule is called pGal. pGal. By the way, some people refer to that molecule right there as PGA as well. Okay, so this one is referred to as pGal, or diphosphoglycerate, because now it's got 2 phosphate groups sitting in it. So this is like a charged version of the 3PG molecule. And then since you made 2 of them, this whole thing is actually happening twice. Even though the drawing is not showing to you, this whole thing is actually happening twice, which means it was going to take 2 ATP to make 2 pGal molecules. Alright? So you're keeping track of that so far. Now, as soon as that's over, the trucks produced, the NADPHs produced during the light reaction at the end of the non-cyclic photosynthesis. Remember, this is why you need non-cyclic. Cyclic is not enough because it doesn't make this truck. Now, this truck is going to go ahead and add an extra hydrogen to the molecule. So you see that there was 1, 2, 3, 4 hydrogens. And now an extra hydrogen is going to be added to the molecule and is going to change the conformation of the molecule. But you're also going to get back one of those phosphate groups from this. So you detached a phosphate group from the molecule right here and replace it with a hydrogen group that is being stolen from the truck. So you leave with an unloaded truck in a phosphate group. Now, this unloaded truck is going to go back, and the phosphate group are both going to go back to the thylakoid membranes, where they're going to be used again to recharge ATP, and used again to recharge for the truck to be recharged. So these guys are going to go back to the light reactions. But this molecule that are now produced is called 3-GP, or glyceraldehyde 3-phosphate. Now, this 3-GP molecule is the molecule that I wanted. The whole point of doing this was to make the 3-GP molecule. But remember, since I had 2 split 3C molecules, I had to also use 2 NADPHs, and therefore I'm going to making 2 G3P molecules. All right? So by completing this half of the cycle, so I mean halfway through the circle here, by completing this halfway to the circle, I actually use 2 ATP, all right, and I use 2 NADPHs, to charge up, transform Rubisco into a molecule called 3-GP, and I actually make 2 of those molecules. Now I'm going to have to keep going to rebuild Rubisco, all right? Now, here's the deal. To rebuild Rubisco, it's going to take some energy. It's actually going to take 3 ATPs to convert and to rebuild this Rubisco. But in order to do that, to rebuild this very complex Rubisco molecule, I need 5 G3Ps. So I am not going to go this way and rebuild Rubisco unless I have 5 3-GPs. So it's going to be a while before the cell can do this. Now let me explain what I mean by that. So that means after this all is said and done, I only made 2 3-GPs. But in order to rebuild Rubisco, since I need 5, I'm going to need to do this 2 more times. So this part of the cycle has to be done 3 times so that I make a total of, remember, each time you do it, you're going to get 2 3-GPs because you got the molecule split into 3 times. So you're going to make 2 ATPs, G3Ps every time. But you need to do this 3 times to make a total of 6 3-GPs so that 5 of them go this way. So 5 of the 6 will go that way to rebuild Rubisco at the expense of 3 ATPs. Meanwhile, 1 of the 3 GTPs will go out of the cycle, all right? Which is actually what I wanted to make. So what is the overall picture here? If I'm getting 3 times this half circle here, that means I capture 3 carbon dioxides. Since each time I go halfway here, I consume 2 ATPs, it actually takes 6 ATPs to go around this way. Since each time I went halfway, I consumed 2 trucks, it actually takes 6 trucks to do this, to make this happen. And then I will get my 6 3-GPs, 5 will go one way, 1 will go another, and 3 more ATPs will be consumed, which means a total of 9 ATPs will be consumed to make 1 of these 3-GP molecules. So, are you following? 3 plus 6 makes 9 ATPs total, right? Okay. So, the problem is, look at the following, follow the carbons. 1 plus 5 makes 6. 6 split up makes 2 molecules with 3 each. This is all it's doing is adding phosphate, so it's still 3 carbons. All it's doing is removing phosphate and adding hydrogen, so it's still 3 carbons. So when you look at this, what you have is still a 3-carbon molecule. So G3P is still a 3-carbon molecule. The problem is that glucose is a 6-carbon molecule. So in order to make glucose, I have to do this whole thing I just described twice. So that means I have to do this process 3 times to get 6 3GPs. 5 will go one way, 1 will go another, but then I have to do that twice. So that means that we had 9 ATPs to make 1 3GP. So to make 2 3GPs, which is what I need to make glucose, it's going to take 18 ATPs. Total of 18 ATPs. And since I needed 6 trucks, it's going to take 12 trucks, 12 NADH pluses to make this happen. Now, since each water molecule that's split to the non-cyclic photosynthesis ends up charging 1 NADPH truck, I'm actually going to require 12 water molecules just to make this happen. Okay? And so, which can actually make 12 ATPs as well, because each water molecule that goes by powers 1 plastoquinone carrying 2 protons, so that means it's going to make 2 ATPs of energy, so you're only going to make 12 ATP from each from those 12 water molecules. Where do I get the other 6 ATP? From the non-cyclic photosynthesis. So from the cyclic photosynthesis. So it all fits together. 12 ATPs produced during cyclic, 6 produced during non-cyclic, plus the 12 trucks produced at the end of the non-cyclic gives me enough energy to complete 1 carbon fixation cycle. Now notice that all of this is actually happening in 3 phases. The phase 1 is Rubisco connecting to carbon dioxide, which is carbon fixation. Phase 2 is the reduction of that Rubisco and changes of the molecule to create the 3GP. Phase 3 is rebuilding out of 5 3GPs the Rubisco molecule. And the other G3P that's left you use to make macromolecules. So now remember that since you're connecting 3 carbons every time you do this, because you have to do this 3 times, so you're going to collect 3 carbons. Since you have to spin twice, you collect 6 carbons, which is why you end up making a glucose with 6 carbons in it. So that's pretty much how the cycle works. Now, in the second picture here, I actually have this summarized for you. All right, it will take 3 carbon molecules to connect with 3 Rubiscos to make 6 molecules of 3PG, which will make, after ATP, 3 of those P-gal of BPG molecules, which got changed again into 3 GP molecules. But I'm going to need 5 of those to go this way at the expense of 3 ATPs to rebuild Rubisco, and 1 of them is going to go on to become the G3P that I use. But since glucose takes 2 of those to be made, I got to do all of this twice, which means I'm going to need 6, 9 times 2, 18 ATP, 6 times 2, 12 trucks. So it all fits together. Now, another thing that I have to talk about before I finish this is the idea that this 3GP molecule, all right, all it is really is 3 carbons connected to a phosphate group here, all right, and some hydrogens there. Now, that's not much. That doesn't really look like glucose. This is still going to have to undergo some metabolic processes to transform glucose. We don't really teach into detail what happens there, but basically, what's going to happen is the exact reverse of glycolysis, where those 2 3 carbon molecules, which are very similar to pyruvate, are going to be combined together to make 1 glucose molecule. But the other thing you need to know is that G3P molecule can actually make several things. Yes, I can get 2 of those and make glucose, but I can also get that same molecule and make amino acids out of it. I can get that same molecule and make glycerol out of it, which is a monomer for fat. So I can make carbohydrates, I can make proteins, and I can make fat out of this 3GP molecule. So take a step back and think about it. All the amino acids that's in your body, you got out of eating plants, because it's plants that do amino acids. All the glycerol that's inside your body that you use to make fat, you got it off the plants. Which means plants don't do just glucose. They don't just store energy. They do all the monomers. They do amino acids to make proteins. They do glucose to make carbohydrates. And they do glycerol, which is required to make fat. So plants are not just the builders of energy. They're also the storers of all the macromolecules of life. They build the building blocks for the macromolecules of life. So they're very, very important. And since you need these phosphate groups, plants will need nutrients like nitrogen and phosphate to make this work. So the whole process requires nutrients, not just the water and the carbon dioxide, which was involved in photosynthesis. All right? We'll pick it up from here on the next lecture.